Hi, everyone. Thank you so much for joining me today while I talk about modern approaches to species diversity measurements. This review of traditional and updated approaches comes from the paper A Conceptual Guide to Measuring Species Diversity by M. Roswell, J. Dushoff, and R. Winfree, published in 2021 in the Nordic Ecological Society's monthly Oikos journal. I am Lauren Quackenbush, and let's get right into it. We've all probably taken a biology class, maybe microbiology, botany, or rangeland science. All of these disciplines may utilize diversity measurements in studies, so we are all at least familiar with the terms richness, evenness, and possibly even the Shannon or Simpson indexes. These are the traditional diversity measurements that were taught when I was getting my undergrad back in 2007 to 2011. So, when I came back for this graduate program and decided to do research involving microbiome diversity, I had to update my knowledge of best practices. And this paper has definitely contributed to that understanding. Equalizing samples is a process that you typically build into your research design. Equalizing effort could be spending equal time sampling each transect, sampling from plots of equal size, setting out the same number of traps in each plot, etc. Rarefying sample size is straightforward by either sampling an equal number of individuals from a community, or in microbiome research, sequencing data is usually shortened to or rarefied to an equal number of base pairs. In diversity research, two factors determine how well a sample represents the true diversity of a community. First is how hard you look, and second is the species distribution and relative abundances. Equal effort and rarefying sample size addresses the first factor, but not the second. And this highlights the difference between true diversity and sample diversity. What does this mean for diversity measurements based on equal effort sampling? It means that those diversity measurements, especially richness, underestimate community diversity because the samples often include fewer species than the community simply by chance alone. As I mentioned, my graduate research includes microbiome diversity measurements, specifically from the rhizosphere microbiomes of four different rootstock cultivar pairs, as well as a couple control samples from the tractor row. I incorporated a traditional equal effort method by taking three rhizosphere samples from each plot. In the sequencing lab that I sent my samples to, rarefied the sequence length of each sample to a maximum of 301 base pairs and a minimum of 100 base pairs. From this data, 14 samples in all, I'll be comparing their compositions, i.e., their richness and evenness, various alpha diversity measurements such as Shannon diversity and Faith's phylogenetic diversity, and beta diversity measurements like Bray-Curtis, weighted unifrac, and unweighted unifrac. I chose these measurements because they appeared the most frequently in research articles about vineyard rhizosphere microbiomes that were published between 2015 and 2021. Following a review of this Roswell et al. Article, however, I may also include some hill diversity measurements. This paper presents arguments that support using different equalizing and diversity measurement standards based on known shortcomings of the traditional methods. I hesitate to call these updated methods new because they've been around for a long time, but only recently in the last 15 to 20 years have these concepts gained traction in species diversity studies. Coverage, for example, was discovered in the 1940s by the founder of computer science, Alan Turing, but only recently has it been applied to the field of ecology. Similarly, Dr. Mark Oliver Hill introduced his unified method for measuring species diversity in 1973, and despite positive reception at the time, it had to be reintroduced to ecologists by Lou Jost in 2006. And now there's consensus among ecologists that hill diversity is the preferred way to measure not only the species diversity of a community, but also differentiation among communities, functional and phylogenetic diversity, genetic diversity, and evenness. These concepts encompass the needs of my review for determining best practices of measuring species diversity within the grapevine rhizosphere. However, Roswell, Dushoff, and Winfree also include in this paper a discussion of two other standardizing procedures, extrapolation and asymptotic estimators. However, the authors ultimately conclude that there is not enough research on these other methods to support their use over standardizing by coverage. Speaking of coverage, let's talk about what that means. Coverage refers to how much of a sample is known or unknown. In other words, how many species can we identify and how many species can't we identify? The example I use here is if coverage equals 0.95, then that means that 5% of individuals in the sample belong to undetected species. This method therefore accounts for both the amount of sampling, i.e., sampling effort, and gives a better estimate of the true diversity of the community. Through coverage, we recognize that more diverse communities require more sampling in order to be well characterized. And coverage does a good job of describing how well a sample captures the true diversity of the whole community. One of the reasons why ecologists have been willing to accept coverage as a preferable standardizing method is that it mirrors a species accumulation curve, something that ecologists have long used to measure sampling completeness. To refresh all of our memories, the expected slope of the species accumulation curve represents the probability that the next individual sampled will belong to a new species. So you can see how coverage already fits in well with current experimental design and also provides a more robust estimate of true diversity than standardizing by effort or rarefying samples. This is demonstrated in the graphic shown on this slide. If you look at the box labeled B in the upper corner, each different colored line represents a different standardizing method. The black curves are standardized effort, and as you can see, one of the black lines ends before the other because you can't control with effort how much of the community is represented in a sample. The blue vertical lines represent rarefying by size, and as you can see, it just cuts off data arbitrarily without regard to true diversity. Only coverage, which is represented by the red lines, can be controlled by the experimenter. The vertical red lines show where the experimenter equalizes coverage, and in both cases, the proportion of known and unknown species would be the same. That would be comparing at equal coverage. Before we get too far along, let's talk about the underlying math of coverage and hill diversity, which was excellently presented in multiple excerpts by the authors of this paper. The top box on this slide shows how to estimate coverage, represented by the letter C. Let's remember that coverage is a measure of how completely a community has been sampled. And specifically, it estimates the total true relative abundance in the sample. To complete the equation to estimate coverage, one only needs to know three pieces of information. N, the total number of individuals in the sample, F1, the number of species with only one individual present, and F2, the number of species with two individuals present in the sample. This reflects a seemingly contradictory truism in diversity studies, which is that while most species in an ecological community are likely rare, most individuals in the community belong to common species. We'll talk more about rarity in a later slide, but for now, rarity should be understood as a community variable which can be mathematically manipulated. We see that rarity variable in the Hill diversity equation in the lower box. Here, D stands for diversity, S is the number of species, i.e. richness, PI is the proportion of all individuals that belong to species I, RI is the rarity of species I, which can be calculated as the inverse of PI, and L is the exponent that is used to adjust the rarity scale, which we'll talk about soon. This equation is conveniently also the equation for a generalized weighted mean, and we'll see later how we can scale this mean differently to calculate each hill diversity measurement. So, while these equations are straightforward, in microbiome diversity studies, we are often dealing with large data sets. Uh, so, the authors list software packages and functions in R that can be used to calculate these values. For the next 3 slides, I'm going to compare and contrast the traditional diversity measurements with their complementary Hill diversity measurements. One thing to keep in mind about these Hill diversity measurements is that they are unified in several important ways that the traditional methods are not. First of all, they all share the same unit and scale proportionally with species gain and loss. Second, they take as a starting point that both the number and the relative abundance of species are components of diversity that cannot be fully separated. In this slide, I compare the more common sample richness to the Hill diversity measurement of species richness. Roswell et al. Point out that richness is a very flawed measure of diversity because it is sensitive to sampling effort and relative abundance and is strongly influenced by the rarest species, which are precisely the ones we know the least about. In other words, richness as a diversity measurement has high uncertainty. Sample richness is highly sensitive to relative species abundance, but does not take relative abundance into account. The uncertainty of sample richness becomes clear when we think about how sample richness might change with a larger sample size. In other words, relative richness at a low sample size doesn't predict relative richness at a larger sample size. How does species richness deal with these shortcomings? First, let's acknowledge that standardizing samples by coverage before calculating richness would go a long way towards informing our knowledge of the true diversity of the community. Species richness still gives high leverage to rare species, just like sample richness, and does this by taking the arithmetic mean of species rarity. Because of this high sensitivity to rare species, species richness can be difficult to estimate accurately outside of experimental settings. So it's best to reserve this measurement for use when the experimental community is completely known or when parameterizing species distribution or occupancy models. The traditional diversity measurements, Shannon index and Simpson index, improve upon richness by explicitly including relative abundance in their calculations, making them more robust estimates of diversity than richness. However, criticism of the Shannon and Simpson indices can be summarized in 2 shared points. First, the 2 measurements have different units, so one cannot be easily compared to or transformed into the other. And second, neither index scales intuitively with species gains or losses. The figure on this slide demonstrates that. You can see how, starting with an even community of 30 species at the top, that when that community is reduced by 10 species, there is a 12% decrease in the Shannon index value and a 2% decrease in the Simpson value. It gets weirder when the community is reduced by another 10 species in the bottom box, and the Shannon index adjusts with a 32% decrease, while the Simpson index adjusts with a 7% decrease. These criticisms are central to why Hill diversity measurements are gaining in popularity. In contrast to the traditional Shannon and Simpson indices, their Hill diversity complements have the same units, and if some proportion of a community species were randomly removed, all the Hill diversities would decrease by that proportion. The primary difference between these 2 measurements is how they scale rarity, and I'll expand on that in the next 2 slides. I explained earlier that the variable L essentially adjusts the rarity scale, and in Hill-Simpson diversity, L is equal to -1. Functionally, what this does is gives high leverage to common species, and therefore, as our sample size increases, the Hill-Simpson value doesn't change too much. Because common species are easy to identify, Hill-Simpson can be estimated with little bias. But because the focus is only on those common species, The utility of the Hill-Simpson diversity is in research that focuses on the relative abundances of common species. In comparison, Hill-Shannon diversity may be that just right measurement. With the L variable equal to 0, it gives equal leverage to both rare and common species. The mean rarity scale for Hill-Shannon is logarithmic, and because species abundance distributions are often log normal, the Hill-Shannon measurement may give the most accurate estimate of true diversity. This measurement can be used in many capacities, including to characterize gradients in biodiversity, which is essentially what I'm trying to do by comparing the rhizosphere microbiomes. In this final calculation slide, I just wanted to demonstrate how each Hill diversity calculation is a transformation of the earlier Hill calculation. That earlier calculation is copied at the top here. And remember that D equals diversity. And also notice that both Hill-Shannon and Hill-Simpson also calculate a total for the variable D. Species richness, when calculated for D, is equal to S, to variable S, because it is a measure of frequency. The other two diversity equations can be found by setting the L values equal to 0 for Hill-Shannon and -1 for Hill-Simpson. You can see the natural log equation in the Hill-Shannon that gives it the logarithmic mean rarity scale, and the reciprocal equation in the Hill-Simpson that gives it the harmonic mean rarity scale. And again, all these calculations can be found using species frequency values and by selecting the rarity scale that you want to use. Very logical, but also difficult to do with large data sets. And so again, the authors suggest different R packages and functions that can be used to calculate these values. We've already talked quite a bit about rarity scales, but now let's actually visualize what we mean when we talk about them. Rarity can be simply defined as the reciprocal value of relative abundance. Here is obviously where species relative abundance is built into the framework of Hill diversity. I mentioned earlier how each Hill diversity calculates a different mean species rarity, because this is a generalized weighted mean equation, and that results in leveraging rare or common species more. You can see in this figure from left to right how species richness, Hill-Shannon, and Hill-Simpson scale species rarity differently. For each of these three histograms, the same data was used. So this visualization is a clean comparison of how the same community can have different diversity values based on the rarity scale that is used. Finally, let's put it all together. The equalizing by effort, size, or coverage, and all three Hill diversity measurements. This figure shows mean values with 95% confidence intervals. And like the last figure we looked at, this data belongs to the same sampled community. So we can cleanly compare how these diversity values will change with different equalizing methods and L values. The biggest takeaways here are that standardization method matters the most when measuring richness. As you can see, quite a few differences between the top three squares, but standardization matters the least when leverage is given to common species. As you can see in the bottom three squares, the Hill-Simpson diversity values. Fairly uniform results in those bottom three squares. So, which Hill diversity measurement should you use for your research? Roswell, Dushoff, and Winfree explain that using more than one diversity metric actually portrays the diversities of communities most fully because it illuminates the circumstances under which communities are most diverse when leverage is given to rare or common species. This, in turn, can be used to describe evenness and dominance and to fully describe the shape of the species abundance distribution. In conclusion, the authors of this paper, as well as other cited ecologists, suggest a robust approach of calculating the Hill diversity profiles for L values from -2 to 1. That concludes the content I've prepared for you today. Thank you so much for listening. I hope that was informative or at least a little bit interesting. If you have any questions or comments, please feel free to reach out to me in the comments or via email. Thanks again.